Good afternoon, everybody. And it's a first of all, thank you for those of you who took the courage to come in person. I know we are slowly opening up our in-person meetings, and so this is a hybrid mode. We have enough people in here who have come in person. We also have a sizable population online. So welcome to both sets of the people, both online and offline. So today it's my pleasure to introduce Samatha Matthews. Many of you, she's quite popular here, so she doesn't need an introduction, but it's customary for.
Sridhar, you're not clear. Sridhar, you're not very clear.
Okay, am I clear now?
Yeah, it is. Yes, okay.
Okay, so it's customary for the supervisor to formally introduce the student. In this case, it is Samatha Matthews. Samatha has a very illustrious student career right from her schooling days, which happened to be at Kendriya Vidyalaya, both in Trivandrum and Chennai. Then took up interest in biotechnology at Kerala University. From there, she moved to IIT Kharagpur for her MTech. And then she was a recipient of this prestigious DAD scholarship that took her to Germany. And from Germany, she decided to come to Delhi. And I still remember the first interview that I was, where Samatha was being recruited. She told us she will be here for exactly 4 months. Okay, we know how that went. She has been with us for quite some time since that time. So she joined as a project assistant in 2014, and it took her a while for me to convince her to join the PhD program. Though from the day one I was convinced that she was going to be an excellent PhD student, but she was not convinced that the lab was right, or probably IGIB was even the right place, or India was the right place. She had a lot of questions on it, but finally we convinced her to stay back with us. In 2016, she formally enrolled, and so she enrolled in a program that involved studying long non-coding RNAs and how they regulate vascular functions. But her contributions to science in terms of our lab and IGIB has been beyond her own PhD, which is something that actually makes a very well-rounded scientist and a researcher. She very actively participated in the rare disease programs that we were doing in the labs. Several of our colleagues were doing. She actually co-authored along with Anjali one of the largest, I would say, scientific policy implementation documents called the Genomics for Rare Disease, the experiences from India, which included about 270 plus authors. That was coordinated by Samatha, and thank you, Samatha, for doing that, because that was not really part of her PhD, but she did do that for the rare disease patients and for the research of the rare disease. That is, I think, a selfless service. Thank you. She also, one more remarkable thing that many of you know, but for the record, I would like to put it on. She has been the go-to person to feed on every holiday in the last 6 years that we have had for the zebrafish animal house. Okay? By default, she has been the person who should make sure that the lowest voices in the lab, which is a technical person, got those days off during the Holi's, the Diwali's, the Christmases, she would be the person who would stay back to feed them. And that, again, thank you for setting examples. And that actually spilled over in the COVID space. In the COVID space for 6 months, I know many of us are stuck at home, some of us are stuck doing COVID genomics work or some other activity around COVID, but Samatha single-handedly, along with Surabhi, am I right? Yeah, along with Surabhi from Shobik's lab, really took charge of the zebrafish lab and made sure that nothing went down in 6 months, and we got back the lab exactly how we had left it in March 2020. For almost, that's a remarkable achievement. Again, thank you for that, Samatha. And yeah, I can summarize as a very good teacher, an advocate for many of you know me. I'm a little bit hypersensitive, and I don't want to praise myself, but I'm not greatly popular in the lab either. So somehow would be the person to come and confront me and tell me that probably I need to take a little bit milder stance, and I would normally yield to that. So thank you for maintaining the balance in the lab. And now coming back to her PhD, she worked on long non-coding RNAs and how they regulate vascular biology. She led, along with Paras, one of the most elegant piece of work that we had done in the lab, which was published a little while ago in the EMBO journal. A fantastic piece of work, and thank you for really taking that leadership to do that. Her own work has spun from that particular observation into a second LNC RNA and how we have traveled that path. And really, we have learned a lot of things in terms of biology, and Samatha will share more with you. Samatha, the stage is yours.
Thank you so much, sir, for that very generous and thought out introduction. Thanks everyone for joining, both offline and online.
A little bit closer to that mic.
So I'll share my screen. So I have worked on the role of long non-coding RNA in maintaining vascular integrity in zebrafish for my PhD project. And I started this work in February 2016. So it's been almost 6 years, including the pandemic period. So what you are seeing on the left is the cranial vasculature of zebrafish. So these are the two eyes, and what you're seeing is an image from the top. This is a double transgenic zebrafish where the endothelial cells are labeled with GFP and the blood cells are labeled by RFP. So wherever there's a co-localization, you can see that there is yellow in color. And you can see here that there's a blotch of red, which means that there are red blood cells outside the vessels and there is a hemorrhage here. And this will be an image I'll be showing throughout. And the theme of my talk will be vascular integrity. So before going into my work, I'll just give a brief introduction. So before First World War, Germany and US had a fallout, and Germany decided to set up a lot of dye factories. And some of the people who were really happy about this were medical researchers, because they could use these chemicals for therapeutics. And you would have even heard about dyes such as Bayer's and Haste. So this is an excerpt from the book, The Emperor of All Maladies by Dr. Siddhartha Mukherjee. And it talks about a medical researcher, Dr. Paul Ehrlich, who got a Nobel Prize in Physiology or Medicine in 1908 for the discovery of a dye molecule which he repurposed against the microbial agent for syphilis called as salvarsan. But he also introduced the concept of magic bullets, which now we would call as chemotherapy. So basically, he was a person who loved to work with these dyes. And throughout his illustrious career, he had tested out different things. And one of them was the method of in vivo staining. So basically what he did was he injected dyes into the bloodstream of laboratory animals. And then he observed that different tissues take up dyes differently. So his conclusion was that this is because the different tissues take up oxygen differently, because these were acidic or basic dyes. Now, his contemporaries did not agree. They thought it was the blood vessels which were actually taking the dyes to different organs which were different. And he famously disagreed that he cannot accept that the vascular endothelium, which lines the blood vessels, can be different in different organs such as liver or the brain. So in spite of being the first person who did the experiment to show that there is a difference in vascular permeability, he did not interpret his results right. And in the next 100 years of research, now we know that he was actually wrong. And in fact, it was his own student, Edwin Goldman, who did repeat his experiment. So in basically he injected the trypan blue dye into the bloodstream, and it could travel to the different body parts except the brain. And in an reverse experiment, he injected it into the brain, but it wouldn't go into the other body parts. So this meant that there is a barrier, and this barrier is called as a vascular endothelium. So the vascular endothelial permeability is what decides the blood vessel integrity as well. So as we all know, the blood system is divided into arteries and veins, and the points where they contact are found by the capillaries. And if you take a cross section of this, the lining of the blood vessel is made with individual endothelial cells. Now, Cliche and Welch et al. Have beautifully showed how in different conditions, based on how tightly the endothelial cells are packed, there can be difference in permeability or leakage. So basically, there is a constant level of permeability in a normal settings. But if there is acute inflammation or chronic inflammation, these can change. And especially in the case of something like cancer, the endothelial cells itself change their structure, such as the permeability changes. Now, so in the 100 years of research, now we know that there are different vascular permeability factors, which would include the protein coding players, but there can also be epigenetic factors or even non-coding factors such as microRNA and LNC-RNA. Recently, we had a chance to sort of collate all this into a review, and we hope that it will be out soon. So we have been interested in understanding the long non-coding RNA-based regulation of endothelium, and I'll give a brief introduction to LNC-RNA. So these are transons which are longer than 200 nucleotide in length, and their hallmark is a spatiotemporally restricted expression, meaning that they have much lower and tissue-restricted expression compared to mRNA. And they also have poor sequence conservation. However, they might have conserved structural features which would give them conserved function. Now, when we study an LNC-RNA, it's important to think whether it's actually the RNA or the DNA element giving rise to it, or the transcription process of that LNC-RNA itself, which is giving that function. So here I show, for example, this is something which is involved in eggs inactivation, and it acts at its RNA level, whereas there are LNC-RNA should arise from enhancers where the DNA element itself is functional, or the LNC-RNA transcription can open up the chromatin such that other interactions can occur. So, and when we try to study these LNC-RNAs in different model organisms, and if we want to understand their function in a different organism or even in humans, we need to look at what are the modalities we can check the conservation for. So like I said, the sequence conservation is limited. Within the gene body, you can find short stretches, or the promoter can be conserved. An important approach we look at, and something I'll also be talking about, is synteny-based conservation, where there are a series of genes A, B, C, D in a row, and they are present in the same block across different organisms, and these usually function in the same pathway. And so if you discover an LNC-RNA within this, you would guess that it is also a conserved LNC-RNA and could be performing a function in the same pathway. Now, if you know that two RNAs are orthologs, you can also check for other features such as conserved number or length of exon, and various different factors which can affect the functional conservation, such as histone marks, tissue restricted expression, conserved structural domains, interacting partners, or co-expression patterns. Interestingly, Ulitsky et al. Showed that you can also do a complementation experiment, so that if you knock down or knock out an RNA in one organism and put back an orthologous RNA, and if it can rescue the phenotype, you can prove that that is a conserved RNA. Now, we have been interested in understanding the role of LNC-RNAs in endothelial cell development using the model organism zebrafish. The vascular development process is a very highly conserved process, and it occurs in two waves in zebrafish. So here I have marked the endothelial cells in green. The precursors of endothelial cells arise in the ventral mesoderm, and they get differentiated into endothelial cells, and they migrate to the midline of the embryo and form the primary vessels, dorsal aorta and pericardinal vein. And this process is called as vasculogenesis. And during angiogenesis, the secondary vessels such as segmental arteries and veins arise from the ventral side to the dorsal side, and they form the second phase of the angiogenesis. Now, if we look at the cellular level, this is what happens. The angioblasts get differentiated from the hemangioblasts, and they get differentiated into arterial and venal specifications. And so this is a cross section of the embryo. So they migrate to the midline, and they form the dorsal aorta and the pericardinal vein, and they then give rise to the segmental arteries and segmental veins. And if you look at the molecular players here, there are many transcription factors which decide that a cell becomes an endothelial cell. And then from there, the efferent pathways would decide whether it will become an artery or a vein. And the Notch DLL pathway would decide if how the migration occurs, and the junctional proteins decide how well those connections form. Now, it has been known for many decades now that microRNAs play a role in all of these functions, and especially from our lab, microRNA 142A3P has been shown to regulate the junctional protein V-cadherin. If we look at the zebrafish LNCRNAs known in these processes, one is Taiwan antisense from Ramani Ramachandran's lab, and the other is Wheel2. Like Sir described, we recently published a work where we showed that the LNCRNA Wheel2 can regulate the protein kinase C beta 2 protein, which regulates the junctional proteins. So this work also becomes a basis of my work. So what we basically did in this work was we used a transgenic zebrafish where the endothelial cells are labeled with GFP. We prepared a single cell suspension, and we used a FACS-based approach to sort these cells. And the poly-RNA sequencing was performed on the transcriptomes of these. And using a custom-built in-house pipeline, we predicted the novel LNCRNA, and using a cutoff, we predicted which would be the endothelial enriched LNCRNA. And we selected 6 of them and named them as vascular endothelial associated LNCRNA or VEALS for functional validation. And from this, I selected VEAL6, which is a syntonic LNCRNA, which you would remember is a way to look for conservation, and therefore we were interested in this. So based on this background study, we proposed 3 objectives for my PhD. The first was to identify the role of VEAL6 in endothelial function, to understand the effect of VEAL6 on the downstream gene, it being a syntonic LNCRNA, and to in general understand its mechanistic role, and if we can identify a conserved partner. Now I have to explain to you how the origin of VEAL6 looks like. So this is basically the zebrafish chromosome, where the proteins ETS1 and FLE1 are, and KC and J1 form a synteny block. VEAL6 is present immediately upstream to the FLE1 gene. So in humans, this is how it looks like. The ETS1, FLE1, and KC and J1 form the synteny block, and there are about 5 genes in this stretch. So, and this synteny block is now conserved across different vertebrates. And so the question we asked was, can VEAL6 be a potential conserved regulator of the FLE1 transcription factor? Now, FLE1 is a master regulator of endothelial function. So if you remember this figure, in the transcription factors which decide the endothelial cell fate, one of them is FLE1, and FLE1 is an endothelial thought of as an endothelial marker. And in mice, when FLE1 is knocked out, it causes a cerebral hemorrhage and it causes embryonic lethality. In humans, it's associated with cancers and blood disorders. So this is why we were interested in studying VEAL6. So we started by the RT-PCR validation of the RNA-seq data we had. So we checked if the VEALs are truly endothelial enriched, and we could compare it to the marker FLE1. And we could see that like FLE1 is enriched in endothelial cells, all VEALs are enriched, and VEAL6 is also enriched in endothelial cells. We then looked at the different stages of zebrafish development and see where all VEAL6 is present. So this is the initial, there are 11 developmental stages we looked at. And in the initial stages, we can find that VEAL6 is expressing. Whereas in the adult tissue stages, we looked at 5 different tissue types and we could not identify the expression. So we can say that VEAL6 is more of a developmentally expressing LNCRNA. Now, to assess if it is truly non-protein coding, we designed an experiment of ribosomal pull down. So basically, we create single cell suspension and we cross-link the proteins and RNA complexes, and we pull down using a anti-ribosomal antibody, and we isolate the ribosomal fraction, and we did a endpoint PCR. So basically, we have the lysate input, which is the total transcriptome, and the RPS were pulled down, which is the ribosomal fraction, and then the negative control for the PCR. So we use 2 different protein coding genes as a positive control, beta-actin and FLE1. And you can see that both of them are present both in the whole transcriptome and in the ribosomal pull down fractions, whereas VEAL6 is present only in the whole transcriptome and not in the ribosomal fraction, which means that it does not associate with ribosome and hence is truly non-protein coding. Now we also wanted to look at where within the cell VEAL6 is present, because this is relevant for predicting the function of the LNCRNA. So we did a fractionation using the sucrose cushion method, and we isolated 3 fractions: cytosolic, nucleoplasmic, and chromatin fractions. And we checked using Western blot if this is truly clean fractions. And by RT-PCR, we could show that VEAL6 is actually cytoplasmically enriched LNCRNA. So we were interested, because this is a syntonic LNCRNA and because we were thinking whether it could regulate the downstream gene, we also looked at the locus. So we tried to map histone marks to this region from publicly available data sets, and we could find marks such as 27AC, ME1, and ME3 in such ratios such that it predicts that it could have, the VEAL6 locus could have an enhancer activity We also looked at larger public data sets such as the Danube code data. And we could again find some histone marks around this region, suggesting that Wheel6 DNA element itself could also have a function. So finally, to really check if it has a role, we designed a loss of function experiment where we designed two morphlinos. These are antisense oligonucleotides, and this is a single exonic LNCRNA. So we designed it within the gene body, and we did a dose titration from 0.2 to 0.5 molars, and we looked for if they can cause severe developmental defects or even give us specific phenotypes such as hemorrhage or patterning defects. And based on this, we chose 0.2 millimolar as a concentration. And we injected this into the animals at 1 to 2 cell stage. And then we screened for phenotypes such as vessel patterning defects or hemorrhage or cerebral hemorrhage at around 48 hours post-fertilization. So on the left is the control images. So these are embryos images imaged at 2.5x. At 10x, you can clearly see how the vessels are properly patterned and formed. Once we injected the Wheel6 morphlino, we could find that there were vessel patterning defects. So this suggested that when Wheel6 is targeted, this could lead to vessel patterning defects. And we also looked for the hemorrhage phenotype. So in Wheel6 and A and B morphlinos, we could see that there was some amount of hemorrhage happening, and especially in morphlino A, about 50% animals would show the hemorrhage phenotype. So this is a control animal showing the cranial vasculature. These are the eyes. You are seeing a view from the top. The green labels the blood vessels. The red shows the blood cells within the blood vessels. So when we inject the morphlino, the vessels are deformed, and you can see there's a pooling of blood, which means that there's a vessel integrity issue, and therefore the blood cells are pooling out and forming a cranial hemorrhage. So this meant that Wheel6 has a function endothelial cells, because when we target it, it is leading to an endothelial integrity phenotype. So in summary, Wheel6 is a synthetic single exonic endothelial enriched LNCRNA. Its expression is restricted mostly to the early stages. It does not associate with ribosomes, and its targeting by antisense mediated method would lead to cerebral hemorrhage. Now, for the second objective, to understand its mechanistic role, we wanted to create a stable model. So for this, we use a gene editing tool called TALEN. So TALEN stands for transcription effector-like activator-like effector nucleases. So basically, tails are proteins which can bind to the DNA based on the repeat variable dye residues. They have a specificity to bind to each of the DNA residues. And so when we attach it to a FOC1 molecule, it is a nuclease. And when you have two talons and it can dimerize, it will cut in the DNA. And the cell will then take up with the NHEJ base repair, which will introduce indels and cause a loss of function at that locus. So basically, we designed a 5' end talon. So the idea was that if we disrupt at the 5' end, the RNA structure would change, because at the end itself, we have introduced an indel. So we injected the talon arms. We prepared the IVT of this in vitro transcribed product and injected, and again screened for phenotypes. So what you're seeing here is a O-dinosodin staining, which labels the blood cells. It binds to the blood cells. So in the control, you can see that the blood cells are evenly distributed, whereas in a talon injected animal, again, we could see pooling of blood in the brain, which is similar, or which is recapitulating the morphlino phenotype. And we use a heteroduplex mobility shift based assay to confirm that there is truly an indel formation in this locus. And we generated now a line of Wheel6 edited animals, and we have now developed up to 5 generations. We did the genotyping till 3 generations and show that there is a 9 base pair insertion, and this would lead to a cranial hemorrhage phenotype, even in a heterozygous condition. So what I observed was that this phenotype can be transient. So here I'm showing you an example where this animal has a hemorrhage around the eye, around the brain region, and so these are images taken across time points, and the hemorrhage vanishes within some time point. And this is the same animal's image from a different from the dorsal view. And so I did this imaging for different animals, and so in the control you can see that there is a normal control with no hemorrhage. And in different animals, there are different sizes of the hemorrhage, and it also occurs at different time points, and it also clears at different time points. So I also wanted to capture this at a better resolution. So I use a confocal imaging based method, which is done with the help of Monica. So basically, if you see here, this is where the clot is. And if you keep observing, you can see the clot goes away. This is better seen in this single channel video. So this was captured from during a course of 8 hours, and within 8 hours, I could see that this clot was moving away. So I was interested in understanding if molecularly we can see some effect of Wheel6 on causing this phenotype, and if there is something reverting back, which is why the phenotype goes away. So we try to also do the rescue in these animals. So basically, like I said, the function can be at the RNA, DNA, or transcription level. So our idea was that if you can put back the RNA, can it rescue? So we injected RNA back at 300 and 150 picogram, and we actually saw that there is some toxicity in this. So basically, at this point, we are still working on this experiment to say whether it is truly caused by an RNA or DNA. But surely we know that the DNA is edited, and the editing of the DNA is causing to a phenotype. Now we also wanted to understand what is happening at the FLE1 locus. So we went back and mapped the original data, endothelial cell data, to our locus. And so this was a Wheel6 enzyme which is predicted, and all the known FLE1 transcript isoforms are downstream to Wheel6. But from our data, we could predict also a novel isoform such that Wheel6 lies in the first intron of this. So we were intrigued to understand if this also is changed by the editing of Wheel6. So we designed 3 primers around the FLE1 locus, one for the major transcript FLE1A1, which gives rise to the protein coding isoform, FLE1A2, which is immediately downstream to Wheel6, and FLE1A3, which is now our novel transcript. So basically we did, so we froze animals at 2 different time points, one at 2.5 dpf or 2 dpf, which is 56 to 60 hpf, when it has a phenotype. Like I showed, it has a hemorrhage phenotype, and after the hemorrhage clears, I again froze the embryos, and now we did the RT-PCR analysis on this. So basically, we could see that the novel FLE1 transcript is going up in both stages. Although once I apply the multiple comparison correction here, it goes away the statistical significance. But what we observe is that there is definitely an upregulation trend in the FLE1 A3 transcript, which is the novel transcript. So this was one finding. So this is one way Wheel6 could be regulating FLE1 and giving rise to the phenotype. Next, we also tried to do a whole transcriptome analysis of these animals. So basically, we did the same thing that we created across. We got animals with a hemorrhage phenotype. We waited till the hemorrhage clears. So we took samples from either of these stages, and we had age-matched controls. And this experiment was done with the help of Rahul. The analysis was done by Dr. Ambli. So this was the amount of data generated, and we also tried to do the correlation heat map to see that if there is a huge difference between stages versus the mutant versus the control. And we also were interested in understanding if there are any reverse patterns. So basically, is there something which is going down at 2 days, which is causing a phenotype, and then it goes up at 4 days, so it rescues the phenotype. And so this was also one thing we were interested in. So basically, so this was done by RNA sequencing, and we needed to validate it. So we took 2 approaches. One was to just use a normal fold chain based, whichever is the top differentially regulated or differentially expressing genes in the mutant. So we took about fold change of greater than 5 and those with at least FPKM of 10, and we got 21 genes here. And like I said, we were interested in knowing if there are reverse patterns, and we found 8 of genes. So there was some overlap between both of these numbers. And finally, we took 18 genes for RT-PCR based validation of this. And so I did the validation again at 2 days with phenotype and 4 days after the phenotype clears. So we got 2 genes which were statistically significantly differentially expressed. Of this, only ROBO1 shows a fold change of 2 at 2 days. At 4 days, all of them show there is no differential expression, which is significant. But still, we see that there is a reversal of ROBO1, which is was predicted from the RNA-seq data. So basically, ROBO1 is a candidate which is going down when we are getting a phenotype, and then it goes back up when the phenotype is getting rescued. So what is ROBO1? ROBO1 is roundabout axon guidance receptor homolog 1, which is roundabout 1. And if you look at the expression in zebrafish, it is mostly present in the brain region. And like you can say from the name, it was mostly known as a gene having function in the neurons. But later on in zebrafish itself, the study which showed that it also has a role in the cardiac function. So basically, this is a control. This is a heart field formation image. So basically, there are two from left and right side. The cells have to migrate and come together and form a tube. So in control, it happens at the correct time point, whereas in the ROBO1 Morphlino, which is the targeting of the ROBO1, this migration is defected. So this means that ROBO1 not only has a function in the neurons, but it also has function in the cardiac and the endothelial cell. So the green marks the endocardium, which is a specialized endothelial cell. And there are also other studies which shows that ROBO1 has a role in angiogenesis. So this is a matrixel based experiment of angiogenesis, where what you're seeing here are the vessels which are formed in the controls. When the ROBO1 is targeted, the vessels are not connected properly, so it has a role in angiogenesis. And when we look back to the literature, ROBO1 is a well-studied protein which functions through SLIT1 ROBO1 pathway. And in the CNS, the neurons express SLIT2, and ROBO1 is a membrane protein, which is the receptor for SLIT2. And a very recent and the similar phenotype what we have observed in zebrafish was observed in mouse brain as well, that when the SLIT1, so the SLIT ROBO pathway was disrupted. So ROBO1 could be a really good candidate for us to take up forward. And recently it has been shown that SLIT ROBO pathway acts through the VEGF pathway, which is the vascular endothelial growth factor / vascular permeability factor, which is well known in angiogenesis and vascular permeability and integrity. So we hope that we have now hit a target that when Wheel6 is affected, ROBO1 is somehow changing, and we'd like to take this up in future as a lead. An alternative approach we took was to see if Wheel6 binds to any proteins. For this, we used a pull-down based approach. So basically, again, we prepared a single cell suspension, and we did cross-linking of protein and RNA complexes. And we used antisense probes to pull down the RNA, and we did a LC-MS. So we did this in triplicates, and I used LAG-Z as a control, so that if there are non-specific binding of proteins, it can be removed. And from the Wheel6 triplicates, we identified certain candidates which had some peptide hits, and of these, there were 3 which had some known endothelial function. Unfortunately, there's a limitation in zebrafish, is that all proteins do not have good antibodies. So we did test out some of the human antibodies here, and it did not work. So we need to follow up this work more for validation. So validation of this experiment. Now, the summary of this part is that using talent-mediated approach, we edited Wheel6 locus, and we recapitulated the vascular integrity defects we saw in the Morphlino phenotype. And this cranial hemorrhage is transient, and we show that it can affect the novel FLE1 transcript and the whole transcriptome. We identified differentially expressed genes, of which we think that ROBO1 is the candidate to go forward with. With pull down, we identified certain interacting patterns which we need to validate further. Now, for the third objective of identifying a conserved counterpart, what we did was first look around Wheel6. So we knew that Wheel6 sequence is not conserved. It is not, we tried to hit it in the zebrafish genome itself. We also tried with the human and other vertebrates, and we could not find that it is conserved sequence-wise. But like I said, one of the approaches for looking for sequence conservation is to look around it. So we found a sequence upstream. So this is the FLE1 region, which you can see is clearly conserved. These are the conservation marks. And we found a 140 base pair stretch, which is upstream to Wheel6. And so we asked whether this sequence could have some role. So I designed a CRISPR against this region, and we targeted this. So when we inject and we screen for the F1 animals, we see that we get a similar phenotype, which we saw in Wheel6. So this could suggest that this upstream region has something to do with Wheel6. It could be its promoter, or Wheel6 could be an extended transcript. So this is something we are interested in looking forward to identify if this conserved stretch has something to do with Wheel6. Wheel6 could also be a conserved RNA. Now, going by the classical synteny approach, which is the second approach we took for this objective, was to look for the human LNC RNAs here. So like I explained, Ulitsky et al. Had shown this, that if we can take a human ortholog and put it back in zebrafish, and if it can rescue the phenotype, we can show that that is a conserved counterpart. So here there were 5 LNC RNAs, and because our interest is in endothelial cells, sorry, so this is the coordinates. If you look at the browser, this is the ETS gene and the FLI1 gene here, and these are the different LNC RNAs. And we designed primers against this, and we looked in the endothelial cell line, human umbilical vein endothelial cell lines, which were available, of which only one of the LNC RNAs we could identify from this line. And this is already a well-studied LNC RNA called CSIR. Now, so what we tried to do was amplify this RNA in our lab. So you can see that it is conserved exon-wise in some of the higher vertebrates, but in zebrafish, the exons are not conserved again sequence-wise. So for the project, what we have done is that we have amplified this. So CSIR has 2 isoforms, V1, which is 1.2 kb long, and V2, which is 600 base pair, according to Bolberda et al. So we have isolated this and cloned this element. So we'll be planning to do complementation experiments. So basically, we'll try to put this LNC RNA back into the V6 edited zebrafish and see if it can rescue the phenotype. So in summary, the upstream of V6 sequence could be a conserved stretch, which we'll be looking for if it could have a function. And if the potential synthetic counterpart of CSIR, if we can test out complementation experiments and see if it is truly a counterpart of V6. So for overall summary, for the first objective, basically we identified a chromatin enriched endothelial LNC RNA, and when we target this by morphino-based approach, we get a loss of vascular integrity. We then, for the second objective, use a TALEN-based gene editing to create a stable line in which we saw that the ROBO1 transmembrane protein is actually going down, which is well studied in the angiogenesis pathway and in the vascular permeability. For the third objective, we edited a conserved stretch upstream to V6, and we'll be testing out how this is, and it leads to loss of vascular integrity, as in the case of editing V6. And we'll be looking at how these are correlated. And as another approach, we'll also look for an ortholog LNC RNA by complementation experiments. So the proposed future work would be to do those two things under the third objective. Questions? Yes.
So there's no question in chat box. If you have a question, you can directly ask Samatha.
Yes, sir. So Anand here has a question in the auditorium.
You're fantastic, Samatha. Congratulations.
Thank you.
So, couple of questions, mostly regarding your objective 2.
Okay.
I see that your primary hemorrhage phenotype is in the brain when you knock down V6.
Right.
And you're basically knocking down V6 in the entire embryo, not brain specific, right?
Yes.
So, any specific reason why the hemorrhage phenotype is happening predominantly in the brain and not other blood vessels? Just integrity related issues. That's one. And another related question I found very, very interesting, that you see a reversal of the phenotype, and you worked a lot on trying to understand the molecular mechanisms there also. So what I was thinking, and I don't know much about the processes in zebrafish. Typically, when a hemorrhage happens in most mammalian systems, the process of clearing out of hemorrhage is not as simple as a direct reversal of the process. There are cellular processes which clear the cells out. And do you have any interest or inclination in trying to understand in future what is it that is exactly clearing that hemorrhage out? Especially if it is happening in the brain and there's a blood-brain barrier also. So these are the 2 questions.
Thank you. Thank you for the questions. So the first question. There are many genes which are known in vascular permeability. When you perturb them, you see the phenotype first around this time point of 2 days and in the brain. The major reason would be to say that because the brain vessels are much more intricate and complex. If you had followed the images which I had shown, the body vessels are more straight and like serial connections, whereas in the brain there are multiple complex connections. So this is a hypothesis. That's why all of those genes usually show, or it is more visible also, I guess. So when there's a leakage there, it pulls up there and you can pick it up. So this is common for all genes, all permeability genes. It is seen across this. That is one way to look at it. Another way, it could be a very specific, specifically brain expressing LNCRNA. For example, now we have also identified something, the ROBO1, which is more expressed in the brain. So unless we look at it at that much granularity, right now I cannot comment on why exactly that is happening, but that is the answer to the first question. The second question of clearing. So in 2019, there was a paper which showed the phenomenon of how the hemorrhage clearing happens in zebrafish. And that is done by the macrophages actually infiltrate to the region of that hemorrhage, and they clear out the red blood cells while it is there. And there are lymphatic vessels, new lymphatic vessels which will form around that clot region, and they will sort of form new vessels and fix that pipeline. So that's how at the cellular level that is happening. So that is known for, in general, how the hemorrhage clearance happens. And this is known in other mammals as well. But our question was that, so there they have not shown what, at molecular changes, what could go wrong. They were using chemical-based ablation, which led to the hemorrhage there. So my question was, because V6 is an LNCRNA and these are regulator fine-tuning molecules, can we see that at a molecular level, if I am perturbing something and something is changing, if it could be at a molecular level, if we can see a signal there. So fortunately, we are now seeing a lead from ROBO1. So I think this is something people have not really explored this, but your question was whether it is already known how it happens. So there's a 2019 paper which did a detailed imaging based analysis. Like I've shown you here one image, they have captured it throughout from like stage by stage, how the lymphatic vessels come there and they fix that vascular pathway, and then how the macrophages would take out the RBCs. They have imaged all of that and they have shown that.
Sheetal.
Hi, Samta. Nice work. So my question is actually related to what Anand was asking. So I wanted to know what are the details that take place after a hemorrhage, the repair pathways. So related to that, the transcriptomics that you have performed, is that of only the endothelial cells or is it of the entire fish?
No, so there's a technical limitation there that from the number of embryos we get, we will not be able to sort out the endothelial cells and then also do a transcriptomics at this point. So we have given the entire embryos for the transcriptomics.
So I wonder if that leads to the noise that that suppresses your real signal.
Yeah, so that's why this is just a lead right now. We'll have to follow it up with more experiments.
And the protein pull down that you performed,
yes,
which cells were taken for that?
Again, whole lysate, because of the technical limitation of, so unless we take thousands of embryos at the time and juice them up, we will not have enough raw material to start the experiment with.
Is it possible to culture endothelial cells ex vivo?
So we have not tried that. There are people who have tried to do zebrafish cell culturing. It is again a tedious process. So we wanted to, so usually we, for example, especially in the case of V2, we could show the specific endothelial expression pattern, and then we followed up with these experiments. Right now, I think there is a technical limitation there that we cannot use only endothelial cells and do that. We have not tried.
Have you tried using a human endothelial cell and see if we'll pick
So
up?
that is where the point of conservation comes in, that right now I do not know if V6 has a proper conserved candidate. Once I identify that, we can go ahead and do that, which is what has been done in the case of VN2.
All right. Thanks.
Shravanti.
Hi, Samata. Congratulations. Very nice work. I have maybe one naive question. Maybe I have missed it. And second thing, I'll come to that later. So I just want to know, because this is a developmental defect at a very unique time point, which is recovered later. Is it happening due to altered pressure during the development at that particular point? Is question one. And question two is, a lot of permeability defects are known in chronic diseases also, for example, diabetes and some infectious diseases as well. So is there any relevance of your LNCRNA in those contexts?
Right. So the first question, again, we are all the experiments we do along with controls, where age-matched controls are screened for the same phenotype, and there we have a fixed basal level of phenotypes we see at 5% or lesser. But in our case, we'll see at least 40% or more. So we know that it is a specific phenotype. I am not sure what you asked when you say if it's pressure related.
So in terms of tissue, in terms of, during tissue formation, the mechanics which a particular, especially a brain tissue receives, I was just wondering that is it a result of that?
So that's why I said that controls, we don't see the same thing at the percentages we see in the V6 edited animals. So we wouldn't say that it's a normal developmental thing. Whatever we're saying, that just means that V6 has a role in that developmental process. And for the second question, again, for diseases, for checking what is the role, we'll have to identify a conserved counterpart in humans to be able to comment on that question.
Okay.
Thank you.
Divya.
Hi, Samdha. Very nice talk. So I have one question. So your Wheel6 Alenciane, is it overlapping to the flea isoforms? Any isoform of flea?
So like I showed you,
yeah, I missed that.
Upstream to the, it is upstream to the known or publicly available data set, whatever isoforms are there. This novel isoform we found only in our endothelial cell data set, which means that it could be a very endothelial specific isoform. And it is not present overlapping with exons, it is present in the intron of this isoform.
Intron of the flea isoform, but it is overlapping to the Wheel6.
Yes.
So I have one so is your morpholino or the talon you are making, how are you making sure that it is not affecting this isoform and only affecting the LNC RNA?
So right now we have shown that the isoform is going up in these animals. So so just like we have to show V6 rescue and delineate the RNA versus DNA thing. We'll have to do that for the FLE1 isoform also. That is a follow-up experiment.
Okay, and the FLE1 knockout for all the isoforms, sorry, is there any FLE1 knockout, or you have made a knockdown of FLE1 and seen what is the phenotype for that? Is it the same phenotype for the Wheel6?
Right. So in zebrafish, FLE1 is duplicated. That is one thing. So there are insertion mutations in FLE1 published by Craig et al. So they do not show a phenotype, or specifically the hemorrhage phenotype. So there are two things. Maybe it is because of the duplication event, and the FLE1 ETS transcription factor families have a lot of redundant transcription factors. So that could be one reason. And another reason is simply that maybe the lab never looked at a hemorrhage phenotype. They were specifically interested in the vascular patterning. So what they did was they targeted using morpholino. They saw some vessel patterning when they target the FLE1 transcript using morpholino. But then when they did the insertion mutation, they could not find the same phenotype. So, but they do not comment on if they found hemorrhage or not. So I do not. So that is the literature standpoint that FLE1 insertion mutations do not show any vessel related defects. Is what they say.
Okay, thank you.
Thank you.
Anybody has any questions? In the Audi?
Yes, there's a question.
And my question is follow up to what for the hemorrhage that you see and for the transient pattern that you see. Question is, you have observed hemorrhage and then there is improvement. Did you observe on a long-term follow-up, did the hemorrhage come back? First question. Second, if the hemorrhage is getting cleared by any other pathway, as you already mentioned that so that any organism has their own capability to rescue themselves for any eventuality.
It depends on the level of hemorrhage as well.
I just want to know.
Okay.
So for the hemorrhage component, one is clear. Do you observe neovascular?
Samantha, it's better to repeat the question because we cannot hear from the screen the audio questions.
So the first question was whether I have observed the phenotype over a long period of time, and if I have observed any neovascularization during this time. So the answer to the first question would be that yes, we have observed till 4 or 5 days, till which only it's possible under the microscope, after which we usually shift to the system. I do not know if in the adult hemorrhages are reoccurring. That is something we have not checked. But what I can say is that there are no gross defects. They don't die immediately, or they have a normal lifespan. They also give rise to ample number of progeny. So I wouldn't say that there is any deleterious effect happening later on in the life for these animals. So we would say that it is like a complete reversal of phenotype. Observing neovascularization, at this point, I would say it's a technical limitation. I have not checked it at that level that I would have to image an animal for those many days and continuously track it. It would not stay alive beyond the 8 hours I have captured the data.
So is there anybody asking any question in Audi?
Yes, sir. Yes, there's one more question.
So actually I have 2 doubts.
Yeah.
One is when you knock out wheel cells and after a time you are seeing that the hemorrhage is like going back.
Yes.
One is that apart from other wheel cells, like you had a lot of wheel LNC RNAs, I think 6 in all.
Yes.
So do you see overexpression of any of the other LNC RNAs or a compensation pathway that might be occurring?
So that's why we did the whole transcriptome to see that we'll be able to, if not weeds, if there's any other pathway coming up, we did not see any great differential regulation in any other weeds.
And neither is this region duplicated like for
No.
Okay. And the other doubt is that during your conservation, you had seen that there is a 5' upstream region which is conserved. So when you did a removal of that.
CRISPR targeting, yes.
Yes. So during that, what was the expression of wheel cells?
So these are follow-up experiments we still need to do.
Okay, you are not checking.
No, no.
Okay. Thank you. Thanks, sir.
There are no more questions from the auditorium. Anybody on Zoom needs to ask anything else?
I don't think anything here, at least in terms of hand here in the Zoom either. So
Okay.
we can go to the CSIR 800.
Okay, thank you. So for my CSIR 800, which is a CSIR project for reaching 800 million people in India for empowering Indian masses, I chose the OMGDX project mentored by Dr. Vinod Skaria, which is open medical genetic assays to enable diagnosis. So I standardized the capillary sequencing based screening for that, the PCR for doing for SDHB gene. In the context of hereditary paraganglioma pheochromocytoma type 4. So it is one of the mitochondrial genes, and this causes tumors which release catecholamines, and this has a high rate of malignancy with the type 4. It has autosomal dominant inheritance. It is the worldwide prevalence is 1 to 9 in 1 million, and in India it is reported, however genetic prevalence is unknown. So this was the locus, and this is where my primers are placed. These are the different variants known in this region, the SNPs, and these are the copy number variants. So I designed primers, 8 amplicons against this, and I showed that these could work. I also prepared a patient information booklet along with this, and along with the CSIR 800 report, I have submitted this. These are the list of my publications. Are there any other questions, or shall I go ahead with acknowledgements?
You can go ahead.
Okay. So I gave a very, very long Wednesday forum acknowledgement because I thought people won't be able to join in my PhD acknowledgement. but I don't know, it's fortunately or unfortunately, we have a hybrid mode in which I have people also listening from other places. First person I have to obviously thank is my supervisor, who has given me infinite freedom in the lab. So Paras had a colloquium and he said that he had the freedom to order any reagent he wanted. I had that and more. So I also always run some side experiment at all times. I've been always ordering some primers and running something. He has never questioned that. And apart from that, away from the bench, I've had the freedom to do science outreach, science communication, and so much more, which I'll talk about in my acknowledgement slides. But and like Sir himself said, we have had a lot of disagreements, which I think is common in any student supervisor dynamics. But we also have a lot of common grounds, I think. The first is how he likes to take responsibility of so many things. So recently, one of the new PhD students asked me, "Aapke sir har cheez mein responsibility lena pasand hai kya?" And then I was like, "Yes, probably he does." And then he also, so while I was at IGIB, I had a chance to sort of rediscover my writing skills, not the science, other than that. And I was so lucky to have IGIB friends who have supported me so much. But all throughout, I would say there is nobody who supports my writing than Sir. I have to admit that. In fact, even before I landed in the lab, the people in the lab were like, "Sir said that there's a girl who is going to come who can write very well, and that's only from my emails." And in fact, when I joined, within 3 months, he had given me 2 first author papers to write, of which one was communicated within 5 months. So that is the level of trust he has shown in me when it comes to writing. And I remember I cleared my CSIR only the second time with the V6 project. And after that, when he realized that I was going to take up V6 for my PhD, he told me that you're taking a risk, because unless this gets a mechanism solved, you will not have anything to show out of your PhD. And if you think back, it is in 2016 where even for V2 we haven't started thinking of how to solve the mechanism. So I remember I just smiled and then I just went ahead with it. He never questioned that again. And apart from all the support and whatever happened during the PhD, I think personally, I have to acknowledge him. There was a time when my personal preferences and my professional preferences were clashing, and I had to step back from some work. He did not ask a question. Let me do that, and it was really, really important for me at that time. Thank you, sir. And one more time point, he talked about the interview in which he interviewed me. So that was again a time I really wanted that job. But I walked into that interview thinking 99% that I won't be selected. But he selected me, and about 3 or 4 years later, he told me that day he had a bet with his colleague while selecting me. And I still don't know if he won the bet or lost the bet. But thank you for betting on me, sir. Thank you so much.
So people who don't know the bet was that she'll pay back for a PhD. When she was saying that she'll go back in 4 months.
Okay. I'll come to my DAC members. First is Vinod sir, again, who is also a mentor, a person whom usually I take some idea to, and he's like, idea to kisi ke bhi pass ho sakta hai. Execution, then I'll shut and I'll go back. So that's what happens with Vinod sir and me. And again, I had the freedom to go up to him and also say a lot of things. Once we argued for 2 hours on text, and finally we were like agree to disagree and went off. But that is the kind of freedom with him also I could go and talk to, and I really consider him as a mentor. And the amount of work he gets done, it's inhuman how much work he can get done in the same 24 hours we all have. That is something really, really inspiring to see how he can get so many things done and coordinate so many things. And one thing which everybody says is health issues. Like, we have gone through so many waves. I don't know how many people appreciate this, but just because he's on campus, we have always just simply picked up the call picked up the phone and called him and asked, sir, what should we do? And that has been such a blessing. I always hoped that I would not reach that state. But last September, I was really unwell and I had to call him multiple times. And at a point, I know I had gotten worried because I wasn't getting better at all. But I am really grateful for him giving all the advice and trying to get me out of that. Thank you, Vinod sir. The second DAC member is Mitali ma'am, who again is a mentor to me. I remember when my DAC committee was getting formed, sir said that you probably need a woman. One of the DAC members has to be a woman. I said, okay, fine. I didn't think much about it, but it came through last year, January. I was in ma'am's cabin. We were talking about something. Customary, she asked me how's work going. And in 5 minutes, I'd broken down. I was crying like anything. And she got shocked. But then she was very kind and she pulled out one of her earliest students' thesis, Dr. Chitra. And she showed me how this thesis is full of negative results, but it is the most beautifully written thesis. So you don't have to worry. Your thesis will get through. So thank you, ma'am, for that. I will talk about ma'am again later in my slides. Probably the least interacted DAC member is Dr. Shantanu Chaudhary, sir. But in my second DAC, he made a comment which I've tried to remember. He said that you've opened so many boxes, who's going to close them? So from there on, I'm not saying I closed any boxes, but I'm just saying that I tried not to open more boxes. So thank you, sir. So I'll thank IGIB. It's the most wonderful place to work at. A lot of people have said this, but I'll also say again, the way we can just walk into any lab and ask for reagents or even discuss things, walk into any PI's cabin and ask comments on your work, that is just so amazing. Thanks to AcSIR for my degree, CSIR for the funding and my CSIR SR fellowship. Directors, Dr. Rajesh Bhokle and Dr. Anurag. I have pelted Dr. Anurag with so many emails. I will send a 4-paragraph email and he will send within 10 minutes, he will send me an email back with 4 points which would have answered everything I said. So thank you so much to Dr. Anurag, and people who are on Twitter know that he has also openly multiple times sort of encouraged me with his comments when I was in doubt. Thanks again to all IGIB faculty. Again, like I said, we can walk into any cabin anytime, ask any question. That is so great, and I'm thankful to all of them. Pratima, of course, every ACSI student bugs her like anything, and I'm really thankful for her presence. And thanks to all admin and HR people who let us do a PhD through processing so many things, or maybe not processing, but okay. Thanks to confocal facility, Manish ji, he has helped us in a lot of imaging, and although I took the training with him, I couldn't continue, but I'm grateful that he taught me so many things. Of course, thanks to Sheetal ma'am's lab and everybody who maintains it, Monica Verma, who dropped her own colloquium work and she back to back, she booked slots with me and got my imagings done. And Dilip, of course, always on ready. Always you can call him for the confocal and he's always ready. I'll come back to both of them again. COVID testing team. Of course, we are doing a PhD in a pandemic, and without the testing, it would not have been possible. I'm really, really grateful to be working alongside people who have put this service mode on for the last 2 years continuously. So happy to be working with them and all the people who have volunteered with the team. I have to thank my fish, because without them it would not have been possible. I have a pair of fish which can give 400 embryos every week, and they have given me for 2 to 3 months. So an average is 50 to 100 embryos per week, but I have a pair which gives me minimum 400 embryos, and it's almost like they know tumhe PhD khatam karna hai, le lo. It's like that they have given me embryos that many, and I've sacrificed so many fishes for this project, so I am really grateful to them. Of course, then which brings me to zebrafish facility members. It is a student-run facility. Unless we all cooperate, it's not possible. So I'm thankful to all the members, especially to Narendra, who is the most sincere person I think we could have had in the fish lab. Even so, the last Diwali and Holi has been here. Holi was last week, and he even offered to do feeding instead of me, but I was like, no, aapko ghar jana chahiye. But and especially because my health was really fluctuating so much in the last few months, I'm really grateful to have him help in the fish lab. Shashi, of course, trained so many of us. Being an in charge, he trained me in a lot of things. And thanks to all the student in charges who take out special time to maintain this facility, and of course, Dr. Chetna, and which will bring me to the lockdown time. I have had infinite freedom again from both Dr. Chetna and Dr. Sridhar. The room numbers 3 and 4 keys were issued in my name for 5 months. It was not given back ever. It was issued in my name and it was in my custody for 5 months. They have had that much trust on me for those months. And I have to thank Surbi. I could not have picked anybody else as my partner during those initial days. She was doing again the end of her PhD, but she will come every day if I ask her. No complaint, nothing. She will come and do the feedings. और कुछ करना है क्या? और कुछ करना है क्या? It's the only thing she would ask me, and I'm so grateful to have such a colleague like her. Jan, Imran, Mohit from our lab. Even though they were completely busy with the COVID testing and the surveillance, they came to the fish lab whenever I called them. Imran and Mohit specifically learned feeding for this thing. And Rohit, of course, to have another in charge is always great. So I had Rohit also as a support. So thanks to all of them. So fish lab is something which runs continuously. You cannot stop it in between. And lockdown doesn't mean that fish will stop eating. But we had to continuously give them procure the feed and water. And Vishu, even while she was at home, she seamlessly made sure that all the vendors are informed at the right time and the things were in place. Narendra again was at home, but he made sure that whenever we called him, he would get back. Paris was busy with COVID things, but he would also be available whenever we called him. I have stayed in IGIB for 7.5 years, which means that I have to thank ISH mess reps and hostel reps. It is my second home. I am looking forward to a lot of great science coming from IGIB in the coming future, because I know that very few people are ready to take up common responsibilities. I want to especially thank Anurag and Meghali, who have been representatives like since forever. Antara also was begging for somebody to just take up mess rep duty from her, and then finally she had to leave it. So, but thanks to 3 of them and all the other in charges, or the representatives who were there before. And I have to thank all the support staff who, I think especially the AC and electrician guys, because I was an in charge, I had to call them, sometimes I had to scold them, but everybody cooperated well. I have to also thank IT people, although they are not here, but and I have to thank Fulvati auntie. She is like a ray of sunshine. I could have the worst day, but if I cross path with her, I have to smile at least once. She'll always talk so nicely, and she loves her job. That is the best thing. She is so sincere at doing her job that I feel like I kya kar rahi hu. So I really like her presence. And of course, Vishuda and canteen staff, although we always complain about them, I have grown on that food, so I have to thank them. Yeah, this is something I have to mention specifically, is I am somebody who is dependent on Twitter. At the beginning of pandemic, that was the heights of me considering quitting my PhD, and I had a lot of people who were ready to act as sounding boards for my doubts and everything, and specifically the Indian Science Twitter people who have talked to me for lengths and discussed, and all the discussions and the kind words and encouragement has really helped me turn around that mentality, I think. Okay, so when I come to work acknowledgement, I have to acknowledge Dr. Paris Segal. So this photo again, I think would show a good representation. Here there are only 2 tables he is occupying. It could be 3 or 4. That's this Paris. And I'll add one more thing which completes this picture. Please don't disturb. If you disturb, it will be danger. That is how working with Paras is. That's how seriously he takes his work. So when I came to Delhi, I probably had a culture shock. I had never been to North India before. I can say that he was my local guardian for 5 years, and he worked so much. I used to think I'm a workaholic, but in front of him, no way to match that. He's a workaholic and an insomniac. So basically, he's running for almost 22 hours a day. And so yeah, somebody saw the wheel to paper and said that their head is reeling from seeing the data. And my response was, he has generated way more data than he has put out publicly anyway. So he has generated that much data, that much work is what he does. And I think even though we have had a lot of disagreements, I have spent with him days, weeks, months, years just discussing the Wheel 2 project, blow by blow analysis of every hypothesis, every experiment, every results. And it was a great time for me to discuss science with him. And that is something that will always have influence on me. Whenever I do science, that how I work with him will always have influence on me, both how I want to do it and how I won't want to do it. The second person, obviously, I have to acknowledge is Amli Shudas. She's a certified superwoman, and you go to her and she'll always have like some 10 jobs running. You go and ask Amli, can you check this thing also? And she'll open one more. And you sit with her, constantly people are coming to her with, can you check this out, Amli, please? Can you help me with this? And she'll just have a smile and she'll help everybody. And you will do some small thing and go to this piece and work. Oh, super cool, so awesome. That's how she'll always respond with that positivity. And it's so amazing to have had her. She actually pointed out the novel transcript, which has been sort of a ray of sunshine in my otherwise dark hypothesis. So thank you, Amli. And again, this is the best image I can find to show Gyan. If he's not doing 10 things at a time, he doesn't feel like he's doing something. So if he's not running 10 gels, he will be running 2 experiments in lab, and he will go to the system and he'll run 4 bioinformatics jobs. So that's how Gyan works. And I think especially in the last 1.5 years, he has been like constant source of support. I haven't ordered one thing. He orders everything and I can just take it from him. And even if I come to lab, I can't find something, first person to call is Gyan. He has also managed a lot of ordering of the lab. So thank you, Gyan, for being there. I have to thank Rahul and NGS team for the transcriptomics essay that we did. And I am now part of a legacy of the non-coding RNA team. I think our lab has different lineages, and right now this lineage is the longest. I am the 4th in this lineage and Gyan will be the 5th. And this work, the Wheels work was started by Dr. Mukesh Lalwani. My interactions with him was just once or twice, but he was always treated me like a teammate, and that is such a great feeling. I also had other great seniors, and so grateful to be a part of all of them. And I have to thank 2 people who have done work for me, for which I have no results to show. First is Ria. So the entire imaging which I showed, I standardized that over 1 year on the light sheet microscope. That microscope was so sensitive that it kept breaking down. I'll call Ria at 12 in the night, 1 in the night, but she'll be so patient. Every time she'll come and fix it with me. Thank you so much, Ria. And after standardizing everything there, I just could go to the confocal switch, and within 3 weeks, I completed the experiment. But before that, all the struggling with the experiment was done along with Ria. So thank you so much. Praveen, again, when the LCMS wasn't showing any great results, I went to him and said, can you check this again? So he worked with me for 1 week. We ran again and we saw sort of got the same amount of hits there also. So I, on principle, have not taken any trainees on my project because I always felt it was a sinking ship. But I had one trainee, Sunaina, whom I trained for 1.5 weeks. But I have also introduced 2 postdocs to the lab. Ankita Mitra is somebody I handed over the technology transfer project. And Dr. Rachna, she was a PhD from Sanjay Gosla's lab. And I introduced her to the zebrafish. Again, I trained for her only for 1 month. Mia Kumla is somebody I trained for a few weeks. but I could understand how good I could be as a mentor, if where all my limitations are and what I'm good at. So I could learn these things by sort of introducing or training them. I have to thank Chetna Ma'am's lab, CSSM, KC, DC, BP labs for sharing reagents at all times. Specifically AcSIR, if I can't close a -80 door, I'll call AcSIR. If I can't find a reagent, I'll call AcSIR. If I can't find a plastic ware, I'll call AcSIR, and he'll always be helpful and happy to help. So thank you, AcSIR. And of course, Kannan, everybody on the first floor have to acknowledge Kannan. Thank you, Kannan, for the cloning expertise and protein. And if I go to him with a question, he will say, sit down, explain the whole experiment. So I'll explain the whole thing, he'll hear the whole thing and then only give me inputs. And I know that you will become a great teacher, but I want to really see that happening, how you can really teach the next generation so well. Thank you, Kannan. I have to thank my true research partners, Google, PubMed, and SciHub for letting me do my work. Now I will come to 8 years of relook into the people I've known in this lab. This is me, if you can't recognize me. So that was 2014, this is 15, this is 17 lab trips. So basically we have had lab parties. This is a reunion when our alumni visited us. This is lab trip. This is when we went for a marriage and made it into a lab trip. And this is a press conference where we were all at. And this is a recent addition of a new member. So I've seen so many moments in 7.5 years in this lab. I have to especially thank Anjali, Vishu, and Srishti for, I think, when everybody else is so just lost in their work, they keep planning things and making celebrations happen. Specifically for me, even though I'm an openly declared atheist, they make sure that all Christmases they do something and make it special for me. Thank you so much. Srishti, I want to again thank for helping me with some of the outreach things. She has helped me with imaging and capturing videos. Thank you, Srishti. I will come to what sir was saying as my extracurricular things. So IGIB Open Days is something I always look forward to for 4 years. This is my how it started, how it's going favorite set of picture. So this is me in 2016 and this is me 2020 giving a talk on the genomics journey of IGIB. In fact, I stood on this stage and talked about the genomics of IGIB alongside faculty. And this is all possible because of the support of only one person, which is Veera ma'am. Every open day after open day, she will always have so many kind words of encouragement to say to me. Once she told me that you don't see yourself like others see you. And that is a set of words I go back to when I'm just lost in my self-doubt. In fact, this year beginning, she came to IGIB. So she drove down to IGIB, listened to my work for 2 to 3 hours, and she drove back. And she, not only on science, she commented, she gave me a lot of advice on many things. And I'm really, really grateful to have someone like her who has said so many kind words to me. I will always keep them in mind. Thank you, ma'am. And of course, I have to thank Pooty and all the science ambassadors for keeping the Josh high and keeping it going. This is my brainchild and personal pride. I don't know if how many people here know about Ignite IGIB think tank. It was something I started for discussing science every month along with my different organizers, and I thank everybody who has been part of it, who took part in it. It was a really great experience for me. I also organize many live of PI events here, and I thank Dr. Ramchandran sir for giving me that opportunity, especially after interacting with Dr. Kang. I mailed her a couple of times. Another person who emails you right back at 6 o'clock in the morning. But it was great to meet so many people. And so when I would basically host them and interact with them, I would read about their histories even before they speak. So that has had a lot of influence on me. Like I said, there have been so many great faculty with whom I could interact. One is TN Vivek. Everybody agrees that he can remember everybody's work. I don't know how he does that. And after my Wednesday forum, he caught me and told me an analogy how PhD is like a pond filled with lotuses, which double every day. So basically, before the day it gets closed, it will only look half full. So this is something he told me in 2019, and I'm happy that finally in 2022 I'm trying to see some closure. But thank you, sir. Every time he sees me, he'll still ask, what happened to that experiment? And that experiment he heard in a Wednesday forum, but he still remembers that. And that's so great when somebody remembers your work so much. Thank you, sir. Dr. Lippy is somebody who herself has always reached out to me and said so many kind words, and that is really had such a positive impact on me. Thank you. Sheetal ma'am is another person who can again remember your work from some talk you gave, and she'll always have such good inputs. She also said some words to me which she may not realize, but had really great positive impact on me. Thank you so much. Kaushik sir, last year in March, he got hold of me and he's like, what work are you doing? I need to hear. And I could give a talk to him. He gave some inputs and some encouraging words. Thank you so much for that. We have also discussed on so many other events I've organized. So that it's been great to have somebody like Kaushik sir to discuss about those things. Again, Dr. Shravanti is somebody for IASF 2020, I interacted with her, but ever since we also interact once in a while. So it's been great to know her as well. One thing which I never predicted I could do in my PhD was write a book. And for this, I have to thank Mitali ma'am, that along with Shreyas and ma'am, I've authored this book, and it's mostly just Shreyas and his brilliance and me being in awe of it. I cannot understand how at the age of 13 or 14, you can have so much knowledge that we can write a book on it. And Mitali ma'am, again, she's somebody who will just stay out of it. Like, you have all the freedom to do whatever you want. And I think for all the discussion we have had on the book, the more she has discussed about my forward trajectory, she is again another person who invests so much time into me. I don't understand why, but she just is so kind. In fact, when the book came out last year, she insisted that Dr. Brahmachari call me. She shared my number, he called me, and for 15-20 minutes, he was just talking, and I was just stunned at the other end and just listening to him. But that is the level of kindness Mitali ma'am has shown to me. And this has also led to a new habit that whenever I sleep late, she will call me in the morning and she'll be like, are you awake now? Because yesterday also it happened. Yesterday I slept at 7:30, she called me at 9 o'clock. And then when I called her back, first question, are you awake now? So now she's also like used to my up and down schedules, which something sir has also accommodated a lot, that he has never asked me what time I'm in the lab. So thank you so much for so many people I've had to guide me, which will bring me to my extracurricular activities of cultures. So I don't know how many people know about Epiphany, but maybe some of you heard. This is a logo designed by me, Vignesh and me. That was the first cultural activity I started with, but then once we started the PhD batch, there was no going back. We of course had also a lot of cultural events, but this was the wall painting we did last time. So Salwa had this ingenious idea of fruit parties. So I am a person, you cannot find me within 1 kilometer radius of a normal party because I get goose. But she had this idea that if you get fruits, nobody will say no to come. So we just cut and eat fruits for 2 to 3 hours. That is fruit party. And it is so much fun. We had that multiple times. And we also did this for Appreciation Day 2017. If you go around the building and see these painted bottles, this is all done by our batch. Sir, please don't laugh. And it is all done in my room. Okay. And we were also the first batch to start the Goonj collection drive as a batch activity. This is us sorting the clothes, before packing it together. And finally, this is the batch which has given me the title of a director. Here I mentioned 3 people, Anurag Raj, without whom I would not be standing here today, having organized all this. I guess you would have seen how much he sort of takes a responsibility for everything. Like in batch, whenever I coordinate anything, first person to call is Anurag. Whenever I need to arrange here a workshop, every first person to call is Anurag. I call him IT head. He is my IT head. But and And even in hostel for the last these many years, I say that there should be a plaque at ISS reception, thanks to Anurag Raj, 2016 to 2022. That is the amount of things he has taken responsibility for from the mess to the AC repair, the painting. I don't know what, everything goes through Anurag. He is like the second director only. And if I could put one word on Anurag, that is reliable. Like you call him, he will be there, he will get it done. That's so great to have such a friend like him. Second is Anjali Bajaj. I would have never guessed that if I have a lab mate who's also a batchmate, how it could change things, because we have a common choice of dancing. Not that I'm great at dancing, but she's a dancer. She likes to teach dancing. I like to dance. So when people joined Cult and went to dance there, we just took our speakers and laptop, and wherever we could connect, we have danced at every nook and corner of this building and that building. So that's how much we have had fun together. So thank you, Anjali, for that. Third is Dilip. So everybody talks about ups and downs of PhD. He is a person who started his PhD from a low, knowingly, deliberately, and from there he has built his PhD back up. And if you heard his colloquium a couple of months back, the DAC members were like heaping praise on him. And that is such a great thing to know about him, how he is so motivated that he keeps doing that. And another thing I have to say about is the confocal slot booking. Like he would have helped hundreds of slots to run, but even now if you call him, his response will be like, okay, great, today I'll get to learn something new. And that is such a great attitude to have, and that is something I also try to emulate. And maybe 4 years back, he told me that if you don't care about your PhD, nobody else will ever care about it. So thank you, Dilip, for being that great friend. I will also come to the musical bands. If people don't know, I sing also. And these are the 3 times I sang. And oh, this is my favorite still. This is me in zombie mode. This was 2019. This could not have happened without Abhilash Gangadharan, who has a moving music studio with whom only we could have done all the practice sessions, and the great Ambli Shivadas, without whose guidance we could not have done any of this, and all the band members. I just love the practice sessions. I don't care that much about the performance. I really love the practice sessions. Thanks everybody for having those fun experiences. Coming to the personal acknowledgements, I think it comes to no surprise to anybody that Monica will be first on the list. This is our first photo together, 2019, my birthday, which was 3 years ago. And then since then, we have had so many photo sessions, and this is one of the most recent ones. I also always tell her that I never thought that I'll find a friend like her in IGIB, but I did. And she is the definition of a friend in need is a friend indeed. So I have only fallen sick, really sick twice in IGIB, both times almost bedridden for 1 whole month. First time I had to go back home to get better. Second time I could get back on feet. People might remember I was not able to walk. I was on painkillers continuously, but there was only one person checking on me every day. That is Monica. She was really busy with her colloquium closing and everything, but every day she would make sure that I get ice packs or medicine or whatever I wanted. Thank you so much, Monica, for being there for when it was the lowest point during my PhD. And I think if I keep talking, I can keep on talking. It's so great to have a friend like her. Again, like I said, the slots, she was so busy, but because she knew that I had to get the confocal slots, she would always plan her work around my slots. She has done that for Meghna as well. I know that. It's so, and how two final year PhDs, both Dilip and Monica, in their ending, they're really ready to plan their work around managing a common facility, something I cannot understand. But thank you to both of them. Second person is, of course, Ambali Shivadas. She is somebody I really look up to. And she is multi-talented, but at the same time, so down to earth. She has everything I want to emulate in my life. Thank you so much, Ambali, for being such a great friend and somebody I can look up to. Arushi Batra, again, I think we have gotten close during the pandemic mostly, but she's a person, an open-minded person with whom I can agree and disagree anytime, and then we can go back to talking normally. So that's great to have Arushi as a friend. And of course, I had to talk about Urmila, who is a person. So IGIB is a place where a lot of people stay away from home. They don't have friends or family to talk to. And Urmila would be that person. I've seen it multiple times that people go to her and say that I have this problem and please advise. And she'll always listen and try to give them advice. And it has happened to me as well. I have told her something I never told somebody for 5 years. And she's been that great person of who has listened and always said words of motivation. Thank you, Urmila. If I talk about other people who have really supported me, one has to be Vignesh. I've known Vignesh since I've been in lab. He joined a few months after me. He's again a person who has a lot of common interests with me. So especially during the lockdown, we used to talk a lot when I was just shut up in this room alone. And he was at home. So thank you, Vignesh, for being a good friend. Jan, again, is a silent support. It's almost like you don't know he is there, and sometimes you realize that if he is not there, there are so many things which go wrong. So thank you, Gyan, for being there. Biplav is a trainee, was in lab, but we bonded during that time, and even now he talks to me, and he's been a great emotional support as well. I have to thank Nishtha, who is a great friend, and she gave me a note last New Year, and is still kept on my working bench, and it's sort of like a source of motivation. Diksha, again, somebody who says a lot of kind words to me and is a good friend. Pratimadi, again, how somebody doesn't have to prove their seniority, and you can just be yourself with anybody in spite of all that, is something I learned from her, and she has also said a lot of kind words to me. Surabhi Rathod from Lipi's lab, again, lot of kind words. Sometimes I don't understand why, but yes. Meghna Arvind, I cannot believe that one person can have so much strength inside them that you look at them and feel like, what are you complaining about? And Meghna has been that source of inspiration for me. She has handled so much alone, and the only thing I can do is just talk to her. It's just unbelievable. There are other people again whom I don't talk to, but I see them every day and think that how are they such fighters, and how can they just keep on smiling and keep on going on every day? And thank you. This is like an informal thank you. I wouldn't ever say to their faces, but every time I pass them, I'm like, you don't have anything to complain about. I have to thank all the musicians, artists, writers, motivational speakers, and all kinds of content creators who push me through the times when I think it's too rough or too dark. I have to thank my long-standing friends and family, friends who don't understand. They're like, what are you doing with your life? But then when they see that I have some academic achievements, they are still there to cheer me, and they tell me they're proud of me. And family, of course, if my parents did not allow me to be here, it's not possible. So this is a photo from last time I went home, 2.5 years ago. I dragged my family out for a weekend getaway at a beach resort, and I think it was very far-sighted of me because then a pandemic happened, and that could have never happened again. My parents, I always say that I don't need inspirational stories because their stories are that inspirational. They were born in extreme poverty. They used education to rise themselves up, and they both retired as gazetted officers from the central government of India. And I have grown up watching their extreme work ethic and discipline, which I will not even try to copy. They're that disciplined, and obviously they look at me and ask, how much time do you need to complete a 5-year degree? But they're that disappointed in me, but I'm trying my best. And my brother, of course, he's the Sant Admi. Like, I don't know somebody who is more calm than my brother in this world. I had a chance to bring him here in 2017. He has seen the fish lab. He has seen the fishes under the microscope. So I discussed with him about fishes, and I can talk about them with science as well. But there is only one person who is responsible for me standing here, which is my mother. This is a photo I clicked last time again. So I put her photo as my wallpaper. But when I went home, I realized she has also put my photo. So I just clicked a photo of that. But it is not easy for a South Indian girl from an average family to be in her 30s, unmarried, away from home, hardly visiting home. And I don't get to know that because my mother takes all the heat and the hits for me. Even yesterday, she called me for something, which is a very important discussion, which I should have as a family member. And I listened to it half and I said, my talk is tomorrow. She said, forget everything, just focus on your talk. And she is a working woman and of course a great role model for me. I find it funny when people say that should people multitask? That's a joke really, because every woman has to multitask, and as a working woman, I've seen her multitask like breathing. So I don't like, for me, it comes normally because I've watched my mother having such a strong role model. I could not have done it in any other way. And I also watched, so I was somebody who used to go to her office after school. So she was only woman in a section of 7 or 8 men, which meant that she had to always know how to vocalize. And although I didn't understand those things at that time, now in retrospect, I think how much she had to fight through that dynamics. And I know that that's what makes me more vocal in some situations. People say that PhD tests your resilience, but in spite of being an extremely resilient person, my mother's resilience was really tested during my PhD and the way I tried to do it. And maybe she has cried as many tears as me, or maybe even more. And I just hope that I can give it back to her. I just hope I get the chance to do that. And so I am standing here only because her deep trust in my decision making that gives me the confidence. And she's truly the wind beneath my wings. So finally, I'll talk about IGIB. I only stayed in one place for maximum 8 years. So in IGIB, I will complete that time soon. So I took a lot of pictures during this time. And this is like a mosaic of all the photos. I've clicked hundreds of pictures, and this is my second home. It's been great to be at this campus, and I have a lot of memories here. I'll just share my favorite quote, which I got from Twitter. The biggest outcome of your PhD is not your thesis, and my own reflection, or your publications, or anything else. It is you. This photo was taken by Asta, and thank you all for patiently listening to me. Thank you.
Thank you, sir. Wonderful. Thank you. Thank you, everybody, for patiently. Very moving. Thank you.
उसमें हो गया नहीं मैंने रोया ही नहीं। I was fully